All right, so as I was saying, before about 1940, scientists didn't think DNA was very important. Before then, they thought that protein was the genetic material. But between the 40s and the 60s, there were several experiments that came along that convinced most people that DNA was really, really important and that they had been missing a step. They knew protein was important. It is. But it's the DNA and the codes for the protein. They didn't know that before about 1950 or so. So yesterday we talked a little bit about one experiment, and we started talking about another one. We talked a little bit about Griffith and Avery, and we talked a little bit about Hershey and Chase. These two sets of experiments are the ones that convinced most scientists that this DNA stuff was really important, that it was the genetic information. So let's review what we talked about with those two here first. Griffith and Avery were working with mice and a particular kind of bacteria, what they call pneumonia bacteria.
Is it a disease pneumonia?
Yeah, it causes pneumonia. There were two strains of this bacteria they were working with, the S strain and the R strain. And the only difference between these two strains of bacteria was that one of them, the S strain, had this capsule on the outside. It was almost like a mucus coating. The R strain didn't have it. But because it had the capsule, the S strain could make things sick. And if you notice, the mice that were injected with the S strain bacteria, they got sick and they died. Whereas the ones with the R strain that didn't have the capsule, the mice got injected with that and they were okay.
So the mucus protects them from the...
Yeah, they didn't really know what was going on here, but we know today that mucus coating kind of helps them to get away from immune systems. Mm-hmm. Next thing that Griffith and Avery did is they killed some of the S strain bacteria with heat. They heated them up really hot, injected those dead bacteria into the mice, no problems. Mice are okay. Dead bacteria shouldn't make you sick. But what Griffith was never able to really explain was the last part of this experiment. They took the dead S strain bacteria, these guys, and mixed them in a syringe with the live R strain. Neither one of these made the mice sick individually. But when you put them in this syringe together, notice the mice died. And when they did like an autopsy on the mice and they grew the bacteria that were in the mice's body, what kinds of bacteria were growing?
R strain live.
Nope.
S strain.
S strain bacteria were growing, okay? They didn't put any live S strain in there. They put dead S strain and live R strain.
The R strain like made that strain live.
So what Griffith was never able to explain, but Avery came along and explained later, was that these living R strain bacteria took DNA up from these dead S strain ones, and it changed them. And it changed them into what? S strain. And we call that a transformation. The importance of this experiment is that what caused those bacteria to change was taking up what? DNA. So that convinced scientists to say, maybe this DNA stuff, maybe it's important, maybe it's the genetic information. Now there were still some people that had some doubts. So the next guys we talked a little bit about yesterday were Hershey and Chase. And they decided to work with viruses. Viruses are kind of the perfect organism to settle this debate, because the only two things most viruses are made out of is DNA and protein, which is the debate in the first place. What's the genetic information, DNA or protein? Here's a picture of what we call a bacteriophage. It's a virus that infects bacteria. Zach, why don't you join us here, buddy? Everything in yellow up here is protein. Everything in purple is DNA. So what Hershey and Chase wanted to do is figure out how this virus attacked bacteria. And was it protein causing the attack, or was it DNA causing the attack? So they had sort of a unique approach. They grew the viruses first in a medium, a food, that had radioactive phosphorus in it. Radioactive phosphorus. It turns out that phosphorus is found in DNA, but it isn't found in what? Proteins. So essentially they made the DNA of the virus is radioactive, but not the protein part. The cool thing about radiation is you can trace it, right? You can tell where it's at. So then they allowed the viruses to attack some bacteria. They grew them together. And afterwards, when they tested to find out where the radiation was, almost all the radiation was where at the end of this experiment? In the bacteria. So what they determined was that viruses make things sick by doing what?
Infecting the
Injecting what into the host cell?
DNA.
The DNA. So again, that proved, at least to Hershey and Chase, that this DNA stuff, it looks like it's the genetic material, because it's what the viruses are injecting. Just to make sure, though, they took another spin on this experiment. This time they grew the viruses in a medium with radioactive sulfur. Sulfur is in protein, but not in what? DNA. So this time they made the protein part of the virus radioactive, but not the DNA. Ran the same experiment. They allowed the viruses to attack the bacteria. Just a second. After the experiment, where was all the radiation at?
And the protein?
Which was where? Where was the protein at the end of this experiment? Nope. Well, the protein was not inside the bacteria. That's what we're going for, right? The protein is not the part of the virus that got injected into the bacteria. The DNA was. So between this experiment, Hershey and Chase, and between Avery and Griffith, most scientists were convinced, hey, this DNA stuff is very important. It's the genetic information. But the weird thing is, now they knew it was the genetic information. What did they know about it?
Nothing.
They didn't know anything about it. They didn't know its shape. They didn't know its structure. They didn't know how it worked. This happened somewhere around 1950, I'm guessing at the date, late 40s, early 50s. So as soon as people were convinced, there sort of started this race, this scientific race, for somebody to figure out the structure of DNA and how it works. And, I don't know about that, but we're talking the 50s. But all the famous biologists sort of jumped on this bandwagon because they knew if you could figure this out, you'd be in every biology, maybe not rich, but you'd end up in every biology textbook ever, okay? Because this is the secret to life, right? DNA is essentially the secret to life. So they are, and we're getting to them. A lot of people tried. Two guys who ended up figuring it out were two guys named Watson and Crick. Watson was this really young guy. He was in his 20s. He's a young American. Crick, on the other hand, was an older British guy. He was a physicist. So you had a biologist and a physicist working together as a team. Watson is kind of a questionable character. Watson's still alive today.
For real?
Yeah. And he still goes around and he tours around America giving speeches about genetics and stuff occasionally. He was in Huntsville maybe 4 or 5 years ago. But he's probably 80 or 85, I don't know, somewhere in that neighborhood. But here's the deal. Watson, after they discovered how DNA works, made a lot of controversial comments. He wrote a book called The Double Helix, which had a lot of controversial comments, talking about how men are superior to women, one race superior to the other race genetically, okay? And that got him in a lot of trouble. He apparently was a really sexist guy in the first place. He didn't think women should be scientists, all that kind of stuff. He eventually, though, became head of the Human Genome Project, which was the project to figure out what every gene in the human body does. And our government spent billions of dollars on this project. Watson was head of it. But eventually, he made some other kind of racially insensitive comments, and he got fired from that job. But he's still out there. He's still pretty influential. Crick is no longer with us. Crick was older when this happened. He was probably already in his 40s when this happened. But Watson's still around. And another couple things about Watson and Crick. Some of the information they got that helped them to figure this out, they sort of cheated. Here's the deal. They knew this woman named Rosalind Franklin. Now we're talking the 1950s. How many women scientists were there in the 1950s? Almost none. There were very few. Rosalind Franklin was working on this project too. She was taking X-ray pictures of DNA to try to figure out the shape of it. Not many people knew how to do that at the time. Apparently Franklin was really good at it. She took some nice pictures. She knew Watson and Crick. She didn't like Watson, because remember, Watson was a sexist. She didn't want to share her research with him, but they had a mutual friend, apparently. Guy was friends with Watson and Crick, and he was friends with Franklin. And somehow, Franklin's pictures got to Watson and Crick without Franklin knowing they got to Watson and Crick. And it turns out that they were one of the key pieces that Watson and Crick needed to figure this out, and they didn't give her any credit. She ended up dying, and really nobody really knew who she was, and they got all the credit. But some other things that happened. There was another guy at this time who was really famous, Linus Pauling. At the time, he was probably the most famous scientist in the world. He figured out how bonds work, like chemical bonds, covalent bonds, figured out how they work. He started working on this project too. But the thing, we're really not still sure how far this went though. His son was friends with Watson and Crick. And a lot of people think that his son was taking information from the father and giving it to Watson and Crick without Mr. Pauling knowing about it, and that they got that information, and that's one of the things that helped them. But that's kind of the way science works. Eventually, though, using information from other people, Watson and Crick never did any experiments. They essentially had this model kit, and they would take information that they got from other people's research and try to build it into a model about how DNA, sort of like what you're going to go do in the lab today. Except they were using like Lego type, not really Legos, but a model kit. And they kept trying to build this model of DNA so that it made sense with all the information they were getting from all these other people. And eventually, it fit. And they figured out essentially the structure of DNA. And once they got the structure, it was pretty obvious how it worked. So in 1953, one of the biggest experiments ever in history, or one of the biggest discoveries ever in history, was the structure and the function of DNA. And Watson and Crick sort of started this field of genetics that we're doing today. And this is eventually going to lead to cures for lots of types of cancer. It's going to lead to ways to treat genetic diseases. It's going to lead to the ability to clone people, clone all kinds of stuff. It's going to be huge. Probably, maybe second to the discovery of antibiotics, this may be the biggest scientific discovery ever. And the two guys who did it were sort of questionable in character, at least Watson was. And they got some information through channels that weren't all necessarily up and up. Let's talk about what they discovered, though. First of all, DNA is in a category of chemicals called nucleic acids. We talked about those early in this class. What are some other nucleic acids besides DNA? RNA and ATP. All those acronym things, right? DNA, RNA, ATP. If you take other biology classes, you'll learn some more. Here's the thing, though. Nucleic acids are built. Remember, most biological molecules, are they big or small molecules? Big. We call them what kind of molecules? Because they're big. What root word means big? Macro. They're called macromolecules. Or sometimes we might call them polymers. And most of these big old biological molecules, even though they're big, they're made of repeating pieces. You might think of my little model here, this thing, as a polymer. But what's each one of these little blocks called? What are polymers made out of? What's the building block of a polymer? Well, slow down for a minute.
Monomers.
They're called monomers. Now, in nucleic acids, DNA is the polymer. It's this big, long molecule made of repeating pieces. But what we want to talk about now, though, are what are the pieces. Each one of these pieces, each one of these monomers, is called a nucleotide. So this is each little building block. Each simple building block of DNA is called a nucleotide. And the pictures you see on the screen right now, these are examples of two nucleotides. Nucleotide on the top and the one on the bottom. You need to know what makes up a nucleotide. In biology, whenever you see rings, like the ones in the middle here, these green rings, those are sugars. Anybody know the name of the sugar that's in DNA?
Ribose.
Deoxyribose. Deoxyribose. That's the name of the sugar. That's here. It's also right here. So in the middle of a nucleotide is always a sugar. And if you notice, there's some numbers around that sugar. 1, 2, 3, 4, 5. What are those numbers numbering? What do you see where there's every number there's a what? What letter? There's a carbon. Okay? They're numbering the carbons. So there's 5 carbons in this sugar. I'll get to that. But just right now, just worry about the carbons. 1, 2, 3, 4, 5 carbons. Attached up to carbon number 1 is always one of these things. These things are called nitrogen bases or nitrogenous bases. Nitrogenous bases, we'll call it. Nitrogenous bases. So there's one here and there's one here. They're hooked to carbon number 1 in the sugar. Attached to carbon 1 in the sugar, the nitrogenous bases. Now, there's several different ones of those in DNA. Anybody know how many different nitrogenous bases there are? There's 4 different ones. And you've heard of this before, because you usually don't think of them by their names, you usually think of them by their letters. What are the 4 letters? A, G, C, T, right? A, G, C, T. Adenine and guanine, which you see up here. Guanine and adenine. And then the other 2 are cytosine and thymine. We'll see those in a minute. AGCT. AGCT. Those are the nitrogenous bases. They are always hooked up to carbon number 1 of the sugar. An RNA that's not in DNA. Adenine and guanine together as a group, they're called purines. And we'll see that when we look at C and T, they look a little bit different. These purines have 2 nitrogen and carbon rings. Notice they're about the same size. The A and the G, very similar in terms of shape. We'll come back to that in a minute. They're called purines. Purines. The other thing we need to talk about, though, that's part of a nucleotide is what's attached up to carbon number 5 of the sugar. Anybody know what you call this thing? It's called a phosphate group. So a nucleotide is made of 3 things. Sugar in the middle. Attached up to carbon number 1 of that sugar is a nitrogenous base. And attached up to carbon number 5 of that sugar is a phosphate group. DNA is made of 2 chains of these things hooked together. 2 long chains of these things. So it's nucleotide, 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 nucleotide. You might think of my little model. If I had 2 of these and they're hooked together in the middle, that'd be what DNA sort of looks like. 2 chains of nucleotides. Now here are the other bases. Again, we've got a nucleotide on top. We've got a nucleotide on the bottom. We've got a sugar in both places. We've got a phosphate group in both places. But notice the nitrogenous bases here look a little bit different, right? They're smaller. This is thymine, and this is cytosine. So we've got T and C. These 2 are called pyrimidines. They're the smaller nitrogenous bases. So you got 2 categories of nitrogen bases. You've got T and C that are sort of small. They're called pyrimidines. They're 1 ring of nitrogens and carbons. And you have A and G, which are bigger, 2 rings of nitrogens and carbons. They're called purines. But regardless, the monomers of DNA, the building blocks of DNA, are called nucleotides. And they're made of sugar, connected to nitrogenous base, also connected to phosphate group. Now, so we can build our models. Let's talk about this and we'll look at a picture, and then we'll go start working on the model. As I said a second ago, DNA is composed of 2 chains of nucleotides. 2 chains. And those two chains of nucleotides then get twisted. You might think of the two chains of nucleotides like a ladder. So here's a chain of nucleotides, and here's a chain of nucleotides. But they're connected in the middle, and we'll talk about what connects them in the middle like this. But it sort of looks like a ladder, right? But then what happens to that ladder is that ladder gets twisted into like a spiral. And we call that a double helix. It's double because it's what? Two chains. And it's a helix because it's twisted. So you want to think of DNA like a twisted ladder. A double helix, a twisted ladder. The rest of this stuff, I'm going to go to a picture to explain. So let's go to the picture first. Here's a nice picture of DNA. What did I just circle right there? That's one way. That whole circle, that's one way. It's a nucleotide. Sugar, nitrogen base, phosphate group. Here's another nucleotide. Sugar, phosphate, nitrogenous base. Here's another nucleotide. Here's another one. Here's another one. And another one. You get the point. DNA is made of two chains of it, left and right chains of nucleotides. Let me get this out of the way. Now, I'm going to call this right here and this right here, I'm going to call those the backbone of DNA for obvious reasons, right? It's like the backbone. What makes up the backbone? What two things make up the backbone in this picture? Phosphate alternating with sugar. It's phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. The backbone of the DNA in both chains, phosphate and then sugar. Sugar, phosphate, sugar, phosphate. Just alternating. Same over here, except going in the other direction. Can that be the important part of DNA?
No.
That is exactly the same in you as it is in a bacteria, as it is in a plant, as it is in a fungus, as it is in DNA viruses. That can't be the important part, right? If it's the same in everything, it's not. Now, I'm not saying it's not important, because it is important because it holds it together, right? That's about support. It's about holding the DNA together. Yeah, go ahead.
You have to have sugar across from sugar.
No, they're running in opposite directions. The important part of the DNA, the part that actually does the coding, are these bases. So notice that you've got each nucleotide has a base attached. If you have a guanine over here, a G, what has to be on the other chain directly across from it?
C.
A C. We say that those two bases are complementary. If we use the word complementary, that means they go together, right? They're a pair. So complementary bases, G always goes with C, and A always goes with T. So if we've got a G over here, we got to have a C over here. If we have a T over here, we got to have an A over here. Now here's the other thing that we need to know. The reason that's the case, what's happening in between these two? What's this supposed to be? And this. Bonds. It's called hydrogen bonds. That's what sticks the two chains together. Now, think for a second. When we were talking about chemistry a while back, what do we keep saying about hydrogen bonds? Well, they form between polar molecules. What do we say about the strength of them? It turns out they're really, really weak. And at first guess, you might think, man, I don't know about that. My DNA is really important. I don't want it held together by really weak stuff. But it is. But it turns out that's a good thing. Because sometimes what does your DNA need to do? It needs to separate so that it can replicate. And so that it can be used to make protein. So it's a good thing that these bonds are breakable. They need to be breakable. But there's bonds between G and C. There's bonds between A and T. Those hold the chain together. The other thing I'll mention, what is the function of DNA? Codes for proteins. And then what do proteins do for you? They build everything. They make your hair, they build your bones, your skin. They make up your enzymes, which make you work, right? So by coding for proteins, DNA pretty much determines everything about you. But the job of DNA is to code for proteins. If you leave here today knowing anything, you better know that. DNA's job, its only real job, code for proteins. Final thing I'll say, and then we'll look at the model kit. Phosphate, sugar, nitrogenous base. That unit is called a what? That unit's called a nucleotide, and it's a monomer, right? You put a bunch of monomers together, you then have a bunch of monomers together, you have a bunch of monomers put together. A polymer, okay? What you're going to try to do today is make a model of DNA. Probably won't finish it today. We'll probably just get started with it. But I'm going to stop the recording here for just a second.